Welcome to the lecture over the cell cycle, which is monitoring cell growth and division. So the human body makes more than 20 billion new cells every day. Over the course of 7 years, all body cells have been replaced except for your nerve cells. So your brain and your nerves will not replace. Now, how do you think your body makes all of these new cells? They make them from existing cells. If you remember cell theory from Unit 1, we know that all cells come from pre-existing cells. And so our current cells will have to replicate and divide in order to create those new cells. Now, what is the purpose then of the cell cycle? The cell cycle regulates the creation of those new cells. And so the division needs to be regulated for those new cells to be created properly. But why do we need to make new cells? There's a lot of reasons why cells need to divide. For unicellular organisms, they reproduce by cell division. So when they divide their cells, they're creating a new organism that is not part of themselves. But for multicellular organisms, we depend on cell division for a variety of things, like development from a fertilized egg. You are much more cells now than you were when you were a fertilized egg in your mother's womb. And so from fertilization, you then developed and reproduced those cells to make new cells by dividing. You also need it for growth. So you make more cells to grow, whether that's growing your muscles, you're making more muscle cells, or just getting bigger and taller. You need to physically create more cells to get bigger, or to repair damaged tissue. So if you got cut, or if you have a wound, you need to create new cells to repair that damage, to replace those damaged cells. So we're first going to review DNA. We talked about DNA last week. There are two types of orientation which DNA can be in. The first orientation is called chromatin, and this is the long and thread-like DNA in a non-dividing cell. And so our DNA, when it is just relaxed, it is in the nucleus, kind of like spaghetti. You can kind of think of it as ramen noodles or spaghetti. It's loose, and it just winds around itself in a loose form. But before our cells divide, this DNA will wrap tightly around histones to create a chromosome, which is this X figure here. It's just this DNA tightly wound to create this X shape. And so chromosomes are thick, short, coiled double DNA in a dividing cell. And so before our cells divide, it has to take it from this chromatin and create this chromosome by winding up tightly to create those X's. Chromosomes are divided into different segments. So you know on the DNA, it's divided into different genes. And so different portions of those genes, or different portions of the DNA, create different genes, whether that's your hair color, your eye color, etc. And so once again, from chromatin to chromosomes, the chromatin is just a loose structure. When it duplicates, it then coils really tightly into this X shape called a chromosome. So once again, what is a cell cycle? It's the repeating sequence of growth and division of a eukaryotic cell. And so it's that regulation of a cell dividing to create a new cell. It is made up of 3 different phases: interphase, mitosis, and cytokinesis. So this diagram down here shows those 3 different phases. We have this very long interphase phase. So it's this very long, most of this circle is interphase. Then you have mitosis, or the mitotic phase, and then right here before it enters interphase again will be cytokinesis. So let's talk about stage 1. Stage 1 is interphase. This is where the cell spends most of its time, and it is split up into 3 different phases within interphase because it is so long. In this phase, the cell will grow, DNA will be loosely wound into chromatin, and the cell will continue to carry out normal functions. But depending on which phase it is in interphase, it does different things. So the first phase of interphase is G1, which we call the first growth phase. And this is where the cell grows and the organelles will start to replicate within the cell. Next, we have the synthesis phase. And this is where DNA replicates to ensure that each daughter cell receives an exact copy of the DNA. And so we talked about DNA replication last week. It is that replication phase of the DNA before it can divide. And then the last phase of interphase is called G2, or second growth. And this is where the cell just continues to grow in size to prepare for division. So you can see here this starting from G1 phase all the way to G2 is interphase. G1, the cell grows and organelles replicate. S, DNA replication occurs. And then G2, it just continues to grow in size to prepare for cell division. Once again, I want to show you the cell is spending most of its time in interphase. About 75% of this phase circle is in interphase. Then we go into mitosis. So you see here the next phase is mitosis. And mitosis is the division of the nucleus. So we're specifically dividing the nucleus into two during this phase. So DNA has already been replicated in that S phase of interphase, but now we need to split this cell into two and divide that nucleus. There's 4 steps within this phase: prophase, metaphase, anaphase, telophase. And we use this acronym PMAT to help us remember the order of which these occur. Stage 3 is then cytokinesis, which is where the cell divides the cytoplasm. So we've already divided the nucleus and we have 2 nuclei now from mitosis. And the final phase is where you're going to physically divide the cytoplasm and create 2 separate cells. In animal cells, the cells pinch in like so, but then in plant cells, it creates a cell plate here to divide them. Here, daughter cells are genetically identical to the parent cells. So, summary of the cell cycle: we have interphase, goes to mitosis, and then cytokinesis divides that cytoplasm, and then it goes right back into interphase because that cell is going to rest until it's ready to move on into the growth phase and reproduce. So now that we've gone through interphase, we're going to go through the individual steps of mitosis. Once again, mitosis is the division of the nucleus into 2 nuclei, each with the same number and kind of chromosomes. This occurs in our somatic cells, which are also known as our body cells. So all of the cells within our body that are not sex cells, egg cells, or sperm cells, then they are going to go through mitosis. A good way to remember this is my toes do mitosis. So just general cells, like your toes would be, is going to go through mitosis, your body cells. There are 4 phases of mitosis. Once again, there's prophase, metaphase, anaphase, telophase. We remember the order by PMAT. You can see visually what's going on in each of these phases. Like prophase, the chromosomes are like so. Metaphase, they meet in the middle. Anaphase, they go away from one another. And telophase, you start to create 2 nuclei for the 2 new cells. So we remember interphase. This is where the cell spends most of its time. There are 3 stages, right? This is what interphase looks like within the cell. You have centrioles over here. You have a nuclear envelope still. The nucleus is still there. And then you have chromatin in the middle of the nucleus. There's not densely condensed into chromosomes yet. But in the first phase of mitosis, the chromosomes will condense to create those chromosomes from the chromatin. The nuclear envelope starts to break down, so the nucleus starts to physically break down, and then these things called spindle fibers will form, which are these little lines that go through between the centrioles, which is going to line up with the chromosomes in just one moment. So 3 steps: chromosomes condensing, nuclear envelope breaks down, and those spindle fibers will form. Then in metaphase, those chromosomes are going to line up in the middle of the cell on those spindle fibers. So they're going to line up in the middle. For anaphase, the chromatids, or the half of the chromosome. So them together make a chromosome, them separated will make a chromatid. They separate and move to opposite poles of the cell. So we think anaphase goes away. Then for telophase, chromosomes are going to start uncoiling, so those chromosomes are going to go back into the chromatid form, like the spaghetti. A new nuclear envelope is then going to form as well, so you're going to get those nucleus back, and now you'll have 2 nucleus from the original 1 nucleus. So you're going to have 2 sets of DNA. If you remember, the DNA has already been replicated before mitosis begins, so even though they separate here, it's still a full set of DNA, because in this form it has twice as much DNA because it's already been replicated. So we have 2 new cells with a full set of DNA. Then cytokinesis is the division of the cytoplasm and the organelles. So the organelles are going to divide into the 2 separate cells, and the cytoplasm is going to be split into 2 to create 2 new cells. And then after that, each of those cells are going to go back in interphase. They might rest in a phase that we call G0, which is the resting phase. But then when the cell wants to start dividing, it's going to go G1, S, G2, and then back into mitosis. So here's another visual. These are cells that physically are in those stages. So we have the interphase here. You can see prophase here, where the chromosomes are condensing and there's no more nucleus. The nucleus is now gone. Metaphase here, they're lining up in the center of the cell. Anaphase, they're starting to get pulled apart into opposite poles of the cell. Then you will see telophase here, where you're going to see those chromosomes starting to uncoil just a little bit. Nuclear envelope is going to start to form, and then cytokinesis is when they're going to pull apart and create 2 new cells. So what is the end result of mitosis and cytokinesis? After mitosis and cytokinesis, the 2 new cells that are formed have the same number and kind of chromosomes as the original. So the original cell we call the parent cell, which would be this one, and the cells formed are known as the daughter cells. So you have the original cell here, DNA replicates, and they go through mitosis and they split and they create what we call 2 daughter cells. Really quick before we end today, I'm going to talk about binary fission, because we talked about how the different types of organisms were replicating for different reasons. So unicellular organisms go through a process called binary fission. So bacteria, they will use cell division to replicate and divide, but we call it binary fission because it is creating a whole new organism. And this is a form of asexual reproduction. It goes in a series of steps. So the bacterial chromosome is the circular DNA molecule in here. So in bacteria, it's in a circular form. The first thing that happens is the DNA replicates. The cell will then double in size, and the cell membrane will start to form in between the 2 chromosomes, and then finally 2 identical daughter cells will result, which creates 2 full organisms. Instead of just another cell in the organism, it's creating a whole new organism. Next, chromosome number. The number of chromosomes each organism has in its cells varies from species to species. So for humans, we have 46 chromosomes. Lettuce only has 18, while chimpanzees have 48. So we all have different numbers or amounts of chromosomes. But all cells, except the sex cells, in an organism will have the same number of chromosomes. So an example for us is we have 46 chromosomes in our cells. So our skin cells, our muscle cells, our heart cells are all going to have a full set of 46 chromosomes, no matter what cell it is in your body, as long as it is not a sex cell like sperm or an egg.